Hi, this is Tiffany Tulo. Week 4, Bio 225. This is my second lab for week 4, biochemical testing for microbial identification. I'm going to be beginning the inoculation part right now, and then I'll let it incubate for 48 to 72 hours. As you can see, I have all my supplies pulled out, and I am ready to begin inoculating. So first, I'm going to be inoculating or pipetting the E. coli culture. Always to sterilize the end. Here is my pipette. You can see it's been sitting in alcohol over here, right here. I'm shaking it dry. Sure. Sterilize. And next I'm going to sterilize the end of my E. coli broth that's been in my incubation area for quite some time now, saving it for other experiments. Going to sterilize that cap, and then I'm going to take a small amount from here to then put in 4 drops into the broth. I'm going to recap the E. coli broth back in my incubation area, and then the MRVP broth that has just had the E. coli placed inside of it will also be recapped and place in incubation area. Do the same here for my S. epidermis broth. Getting it ready here. It's the other already prepared culture. That one as well. Shaking out my pipette. Going to take a tiny amount from here, and transferring it into the MRVP broth. Put this in bleach. Going to flame this. To recap. Now flame the MRVP to recap also. I'll place them in the incubation area for 48 hours, and I'll come back to them and check them out. It has been approximately between 40 and 72 hours. I'm going to be performing the second part of the methyl red test, catalase testing. I've gathered all my supplies, and let's see, I have all my supplies. Here's my reagents, my test tubes. I'll be pouring them. I've been sterilized those with alcohol, and I'll be transferring them in so that I can add the drops of the methyl red reagent to test to see what happens. I'm now going to add the methyl red reagent to, first we'll do my E. coli broth, or I'm sorry, S. epi broth. We'll do 7 drops. Let's see. 1, 2, 4, 5, 6, 7. Let's see, turn red. Next we'll do the E. coli broth. 1, 2, 3, 4, 5, 6, 7. Turned red also. Move this down on a paper towel. And they appear to both test positive. They're both red, not the yellow color as explained in our lab. So now to the Vosges Pro Scour test. I have Barrett's test reagent A. I'm going to add 12 drops to the remaining test tubes. We're going to start with E. coli first. So we're going to do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then the S-epi tube. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Put that out on a paper towel. And I'm going to swirl each of these a little bit gently. Gonna take a look, they're both cloudy. And then we're going to take the reagent B. And you can see, and I'm going to add 4 drops. So this is the E. coli. 1, 2, 3, 4. And the S-epi. 1, 2, 3, 4. I'm going to swirl these for about 30 seconds to expose the contents to the oxygen, and then I'm let them sit for 30 minutes. Now I'm going to be doing the catalase testing. I have my slides labeled, as you can see here. My inoculation loops have been sitting in alcohol. So first let's do the E. coli. Here's my dish. I'm going to scrape some cultures off of here very carefully and place the lid. I'm going to now smear it onto my slide. I'll be placing this in bleach over here. I'm going to do the same with this epidermis culture. See this epidermis is growing even more. Take some of this, gently scraping. There's a very strong odor coming from here now. Place the lid and smear it on my slide. And over in the bleach. Now I have my peroxide in a cup. I'm going to take some of the peroxide and do 1 drop. 1 to the samples. So this is my boy sample. We'll do 1 drop here. There are bubbles present, forming, which you can kind of see there. Definitely bubbles, which indicates a positive result. And the S-epi, we'll do a drop here, which there are definitely bubbles forming as well, which also indicates a positive result. I'm gonna record these and then record them into my lab.